Hi, welcome to Keynes on Science. This is a series of test yourself questions for biology revision on the topic of digestion. Let's dive straight in. Question 1. Which organ is the stomach? A, B, C or D? Stomach is C. Question 2. What type of enzyme is produced in the stomach? Carbohydrates, lipase or protease? Protease. Question 3. Which organ is the small intestine? A, B, C or D? It's D. Question 4. What type of enzyme is produced in the salivary glands? Carbohydrates or amylase, lipase or protease? It's carbohydrates or amylase. Question 5. Amylase is an enzyme that digests starch. Which organs in the human digestive system produce amylase? Is it liver, small intestine and large intestine, salivary glands, stomach and liver, salivary glands, pancreas and small intestine, or stomach, pancreas and large intestine? Amylase is made in the salivary glands, pancreas and small intestine. And 6. Protease is an enzyme that digests protein. Which organs in the human digestive system produce protease? Is it liver, small intestine and large intestine, or salivary glands, stomach and liver, or stomach, pancreas and small intestine, or stomach, pancreas and large intestine? Protease is made in the stomach, pancreas and small intestine. Question 7. Which organ is the liver? A, B, C or D? It's B. Question 8. Which organ produces bile? Large intestine, liver, mouth or pancreas? Bile is made in the liver. Which term describes the pH in the stomach? Is it acidic, alkaline or neutral? Stomach contains hydrochloric acid, so it's acidic. Question 10. How does bile help in the digestion of foods? Does it increase the surface area of fats? Does it an enzyme that digests protein? It makes the pH in small intestine acidic, or it neutralises the stomach acid? Bile increases the surface area of fats and neutralises the stomach acid. 11. How does bile emulsifying fat speed up digestion? Does it increase the surface area of the fat? Does it act like an enzyme that digests the fat? Does it make the pH in the small intestine acidic? Or does it cause the small intestine to absorb fat? Increases the surface area of fats, therefore quicker enzyme action. 12. Where is protein digested in the human digestive system? Is it large intestine, liver, salivary glands, or stomach? Protein is digested in the stomach. 13. Where is amylase made in the human digestive system? Is it the large intestine, the liver, salivary glands, or the stomach? Amylase is made in the salivary glands. 14. What is starch broken down to in the human digestive system? Is it broken down to glucose, fatty acids, amino acids, or glycerol? Starch is broken down into glucose. Question 15. What is protein broken down to in the human digestive system? Is it broken down to glucose, fatty acids, amino acids, or glycerol? Protein is broken down into amino acids. Question 16. What are fats broken down to in the human digestive system? Fats are broken down to fatty acids and glycerol. Question 17. Where is bile made in the human digestive system? Is it made in the large intestine, the liver, salivary glands, or the stomach? Bile is made in the liver. 18. Amylase digests starch in the mouth. What is the optimum pH for amylase? Is it pH 2, pH 7, pH 8, or pH 14? Amylase works best in the mouth at pH 7. Question 19. Protease digests protein in the stomach. What is the optimum pH for protease enzyme? Is it pH 2, pH 7, pH 8, or pH 14? Optimum pH for protease enzyme in the stomach is pH 2. It's why the stomach contains stomach acid. Question 20. Where are most food molecules absorbed? Large intestine, liver, small intestine, or stomach? Most food molecules are absorbed in the small intestine. Question 21. Villi are adapted for efficient absorption of food by having microvilli. How do microvilli help with this function? So, do they maintain a diffusion gradient? Do they increase the surface area? Do they provide a short diffusion pathway? Or do they release energy? Microvilli increase the surface area. Villi in the small intestine are adapted for efficient absorption of food by having many mitochondria. How do mitochondria help with this function? Do they maintain a diffusion gradient? Do they increase the surface area? Do they provide a short diffusion pathway? Or do they release energy? Mitochondria release energy by respiration to allow for active transport in the absorption of food. Question 23. Villi are adapted for efficient absorption of food by having a good blood supply. How does having a good blood supply help with this function? So does a good blood supply maintain the diffusion gradient, increase the surface area, provide a short diffusion pathway, or release energy? Good blood supply maintains diffusion gradient. Question 24. Villi are adapted for efficient absorption of food by thin walls that are 1 cell thick. How does this help with their function? Does it maintain the diffusion gradient, increase surface area, provide short diffusion pathway, or release energy? It provides a short diffusion pathway. There's a very short distance for diffusion to occur. Question 25. Which chemical can be used to test for glucose? Is it Benedict solution, iodine solution, Biuret solution, or ethanol and water? The chemical used to test for glucose is Benedict solution. Question 26. Which chemical could be used to test for protein? Is it Benedict solution, iodine solution, Biuret solution, or ethanol and water? The chemical to test for protein is Biuret solution. Question 27. Which chemical can be used to test for starch? Is it Benedict solution, iodine solution, Biuret solution, or ethanol and water? The chemical used to test for starch is iodine solution. Question 28. Which colour change will be observed when testing for glucose using Benedict solution? Would it be blue-black, or red-orange-green, or lilac, or milky white? Benedict turns red, orange or green from blue when testing for glucose. Question 29. Which colour change will be observed when testing in starch solution using iodine? Would it be blue-black, red-orange-green, lilac or milky white? Iodine solution with starch goes blue-black. Question 30. Which colour change would be observed when testing for protein using Biuret solution? Would it be blue-black, red-orange-green, lilac or milky white? Biuret will turn lilac from blue when testing for protein. Question 31. What chemicals found in the digestive system break down food? Is it stomach acid, bile, enzymes or water? Chemicals that break down food are enzymes. Question 32. What are enzymes made from? Protein, fat or carbohydrate? Enzymes are made from protein. Question 33. What theory describes how enzymes work? Is it digestion theory, diffusion theory, or the lock and key theory? Enzymes work by the lock and key theory. Amylase breaks down starch. This is explained by the lock and key theory. What happens to the active site if the temperature is too high? Does it stay the same, get bigger, get smaller, or change its shape, which we say is denature? The active site changes, which is denature. Hope you found these questions useful. Please subscribe and I'll catch you next time.